announcing the arrival of Professor Dr. Adiba Kamarul Zaman, the Dean of the Faculty of Medicine, University of Malaya, and Professor Dr. Umah Rani Kuppusamy, Department of Biomedical Science, University of Malaya. Ladies and gentlemen, Professor Adiba Kamarul Zaman and Professor Umah Rani Kuppusamy. Thank you. Please be seated. Assalamualaikum and good afternoon. Professor Dr. Adiba, Dean of Faculty of Medicine, Professor Dr. Umah, our distinguished lecturer, distinguished guests, honored family members of the Kuppusamy clan, the management of the Faculty of Medicine, ladies and gentlemen. Welcome to the inaugural lecture by Professor Dr. Umah Rani Kuppusamy, entitled Unravelling the Antioxidant Enigma in Health and Disease. And to begin, I would like to invite Professor Dr. Adiba to chair the lecture and introduce Professor Dr. Umah Rani Kuppusamy.
Assalamualaikum and good afternoon, everyone. Welcome to this afternoon's inaugural lecture by Professor Dr. Umah Rani Kuppusamy, who is currently our head of department of biomedical science. Professor Umah, as she's best known, obtained her honors degree in Bachelor of Science in Biochemistry from University of Malaya in 1989 and PhD also in Biochemistry from NUS in 1994. She started her career as a lecturer in biochemistry at the then International Medical College, currently known as International Medical University, in 1994. And to their loss, but to our faculty's gain, Umah decided to return to the University of Malaya in November 1996. She was promoted to the position of Associate Professor in 2002 and subsequently Professor 8 years later. Prof. Umah has significantly contributed to the teaching, research and administration, particularly in the area of biomedical science. She has been involved in both undergraduate teaching and also postgraduate involvement in PhD degrees and so forth in the biomedical sciences. She has not only contributed to the faculty in the area of biomedical science, but has also been a leader nationally in developing the biomedical science discipline nationally and also regionally. She led a team of academics from various public universities to look into the future direction of biomedical science degree program in Malaysia, and was responsible for establishing links between several universities in Asia, and was amongst those instrumental in forming the Asia International Biomedical Science Consortium. She was in the committee to draw the Allied Health Professions Bill for Biomedical Sciences, which was passed just recently in February of 2016. I think we're still waiting for the enactment of this bill. She was appointed as a member of the Heads of Health Science Council and currently represents UM at the Health Science Dean's Council at the Ministry of Higher Education. On the research front, Umah is considered to be amongst the pioneers in the field of free radical and antioxidant research in Malaysia, and her interest is on fundamental research around free radicals and antioxidants in diseases, and we'll hear more about it in her speech, particularly in the etiology and complications of diabetes and obesity. And this, I'm sure, will be an enormous contribution, considering that diabetes and obesity are probably at the forefront of diseases in Malaysia that we have to deal with. Like many of the academics at the Faculty of Medicine, University of Malaya, Prof. Umah is not only a leader in the academic sense and also in a research sense, but she also contributes enormously as a community leader, having been involved or is still currently involved in several religious organizations and community service organizations. She's a member of the Human and Nature Society, and for her commitment and her services, she was conferred the Ambassador for Peace Award by the Universal Peace Federation in 2009. And since 2010, she has been the Vice President of Women's Federation for World Peace Malaysia. And this organization is involved in organizing seminars that address issues pertaining to women, children, and family peace, as well as peace and charity-driven activities. So we all look forward to hearing Umah's lecture entitled Unravelling the Antioxidant Enigma in Health and Disease. Umah.
Thank you for the kind introduction, Prof. Adiba. Very good evening to Prof. Adiba, the Dean of Faculty of Medicine, honorable guests, ladies and gentlemen. I wish you all a very warm welcome to my inaugural lecture. I hope I'll keep you entertained for the next half an hour or so. Today I'll be sharing with you my research journey on my quest for the unravelment, for the unravelment of antioxidant enigma in health and disease. And I would like to mention here that this lecture is dedicated to all my teachers, including my parents who are my first teachers. Oxidation. Oxidation is actually removal of electron. So when you see nail getting rusty or cut apple turning brown, this process is oxidation process. So an oxidant is an atom or molecule that removes electrons from another atom or molecule during a redox reaction. So free radicals are oxidants, but bear in mind, not all oxidants are free radicals. I'll explain to you why in a short while. And this is the person who actually discovered the first organic radical, and this molecule is known as triphenyl methyl radical. And this discovery was made in 1900. Okay, now coming back to the free radicals. Free radicals are atoms or molecules with a single electron, means usually the electrons have to be in pairs so that they are stable. But in this case, they'll be unpaired, single, and therefore they are very, very unstable and have a high propensity to abstract an electron from other atoms or molecules so that they can stabilize themselves. So when that happens, okay, the molecule, the stable molecule that has lost its electron will become a radical. So instead, it will then go ahead to abstract an electron from another molecule or atom. So this is something like a domino effect. So the chain reaction will go on and on and on, damaging more and more atoms and molecules. So bear in mind there are metals and their ions or complexes which may have unpaired electrons, but they are not classified as radicals because they are relatively stable. Okay, so that's the difference. Now, in our system, we have what you call the reactive oxygen species, as well as the reactive nitrogen species. So these are the group of molecules that may be found in our system. And of course, usually for radicals, it's represented with a dot, which means there's a single unpaired electron. And non-radicals, okay, they are not, technically speaking, they don't have that single electron, but they have a susceptibility, high susceptibility to be converted to radicals, especially when they react with some of the radicals. For an example, you have the hydrogen peroxide, which is classified as non-radical, but it is known as a reactive oxygen species because it has got a high susceptibility to be converted to a very active hydroxyl radical. Now, this hydroxyl radical is known to be one of the most potent radical in our physiological system because it can cause a lot of damage to all the macromolecules in our body. All right, apart from that, hydrogen peroxide is also susceptible to be converted to radical, especially when they are exposed to redox active elements like ferrous and copper. So these elements will participate in a reaction called Fenton-Haber-Weiss reaction, and they can amplify the radical-induced damage. So the radicals are continuously generated, and this can be really a very amplified reaction. Okay, now similarly, you have the reactive nitrogen species. So the main key factor here is the nitrogen molecule, nitrogen atoms. All right, and if you notice, among all these molecules, oxygen seems to be a key player. Okay, for both the reactive oxygen species as well as reactive nitrogen species. Now, having said that, how do we actually get all these radicals in our body? So our normal metabolism actually generates quite a lot of free radicals. So especially in the mitochondria during the electron transportation, during the respiration, okay, when there's electrons transported across the inner mitochondrial membrane, there are superoxides generated. And of course, from the exposure to the environment, you have the UV light that generates free radicals. Then you have the ionizing radiation, smoke, and then all kinds of air pollution, pesticides, chemicals, all these generate radicals. And collectively, these radicals have the tendency to damage your DNA, lipid, proteins, and any other molecules in the body. Fortunately, we are equipped with antioxidant system. So we have what you call the endogenous antioxidants as well as the exogenous antioxidants. So I'll talk about the endogenous antioxidants. These comprise antioxidant enzymes and their coenzymes, glutathione redox methyl ion binding proteins. All these are part and parcel of the endogenous antioxidants. So just to talk about the enzymes, I've given abbreviations, but this is actually superoxide dismutase, which converts a superoxide anion, okay, which is a radical, to hydrogen peroxide. And catalase and glutathione peroxidase, these are enzymes which can inactivate or convert the hydrogen peroxide to water molecules, which are harmless. Now, bear in mind, hydrogen peroxide is commonly produced in our body during normal metabolism, and they can easily diffuse across all compartments of membrane. It could be the plasma membrane or even the mitochondrial membrane, nuclear membrane, any membrane, it can just pass through and potentially cause damage if it's converted to a radical. And of course, we have what you call the glutathione reductase, which this is glutathione reductase, which ensures that the glutathione, which is also known as an antioxidant, is maintained at a reduced state. Okay, now apart from that, we have the exogenous antioxidants comprising vitamins A, C, E, and many phytochemicals, which you can obtain from all the fruits and vegetables, nuts and grains, spices, and also mushrooms. All right, when you are in a healthy state, what happens is your oxidants or free radicals are in balance with the level of your antioxidants. Now, we should not aim to wipe off your the free radicals or oxidants, because that can be detrimental. So in your cellular system, we find that the little bit of free radicals that's there in balance with the antioxidants are required for cell signaling and also to stimulate your endogenous antioxidants so that they are at prime. So whenever there's a free radical, exposure to free radical, they can actually act upon it quickly. Now, unfortunately, let's say you have more oxidants or free radicals, which can happen when you're exposed to pollutants, pesticides, or some toxic chemicals, or even tissue injury, or stress, lack of sleep. So all these can actually increase your level of oxidants and free radicals in the body. So which might be much more than what your endogenous antioxidants can cope. So in that situation, you have a phenomenon called oxidative stress. Now, when you say oxidative stress, the excess free radicals and oxidants here can cause a lot of molecular damage. So which means, like in the cell system, you find your enzymes can be inactivated because of the damage to the proteins, and other transport proteins or receptor proteins can be damaged, and so a lot of cellular functions can be impacted. So therefore, it is not surprising that oxidative stress has been linked to or associated with the etiology of many diseases. Almost every conceivable disease has been linked to oxidative stress. Okay, now let me talk to you about my journey. So this is the storytelling part. Okay, so Prof. Adiba has already mentioned that I obtained my first degree from So I'm a UM product, originally a UM product actually. So in 1989, I obtained my Bachelor of Science in Biochemistry, and then I pursued my postgraduate studies in NUS at the Faculty of Medicine, Department of Biochemistry. Now this is my late professor, Professor N.P. Das. He was actually the pioneer in the field of free radicals, particularly flavonoids, probably in the world as well as in Asia. Sorry, I should say in Asia as well as in the world. All right, so this is our team. So each one of us were working on different aspects of flavonoids. So like this girl was working on food, these two were working on cancer, and this one was on adiposity, or rather lipid. And I was working on enzyme-targeted drug discovery, and this guy here was working on bioavailability of flavonoids. So we had a very good team, and we had weekly discussions to bombard each other with questions, so that we always were on our toes to think about what to do next and whether it's going to be novel or not. And this is the basic structure of flavonoids, the molecule that we have been working on. And this molecule is actually, these are a group of molecules which are found in plants, not in mushrooms or in algae, because mushrooms and algae don't synthesize these. Even if you find, because there are some reports which say that it's found there, but especially in mushrooms, it's absorbed from the material in which the mushrooms grow. So the mushrooms don't synthesize flavonoids. There are thousands of flavonoids. Why the variations? Well, just by substituting all these, the hydroxyl groups and or the sugar groups, so that gives the variations in terms of the structure, and therefore you have different types of flavonoids. I learned a lot while in NUS. My supervisor's mantra was, work hard but don't work like an ant. Don't be penny wise, pound foolish. Don't waste taxpayers' money. So I must share with you some little story here. Within the first 2 weeks, I was working very hard in the lab. I used to work until about 10 o'clock at night. And one day my supervisor dropped by and said, good, you're working until 10 o'clock. So have you got enough data? I would like to see your data. I said, yes, I've got some interesting data, but I can't reproduce them. I've repeated about 3-4 times, but I can't reproduce them. Each time I'm getting a different pattern. To tell you now that I worked on some enzyme activities and 30 different flavonoids. So it's actually quite tedious. You start in the morning and by the time you finish, it's at night. And he uttered this word. He said, I want my students to work hard, but I don't want them to work like an ant. So that actually jolted me. So I said, okay, he's telling me that I'm working like an ant without thinking. So let me think. So I realized, why was I that I couldn't get a reproducible result? First thing, I did a lot of reading up and I found that these flavonoids were unstable in solution. And secondly, found that the solvent that I was told to use, which is ethanol, was not the appropriate solvent. It couldn't actually fully solubilize this flavonoid. So that's when, after a lot of reading up and research, I found that DMSO, okay, was the most suitable solvent for these flavonoids. So, and what I decided was to freshly prepare the flavonoids. The flavonoids are very expensive. So therefore, my supervisor had advised all the students to prepare the stock and aliquot them and store in the fridge and reuse them for each essay. So probably that was another reason why I was not getting reproducible result, because they become inactivated. So I told him after 2 weeks, I told him, look, this is what I discovered, DMSO. All your previous students were wrong. I'm right, DMSO is the solution to use for this. So that was something I learned. Don't just blindly follow whatever your seniors might have done or your publication may say. You have to, when you find that you can't reproduce something, you know obviously something is wrong and you have to make some changes. Well, as for don't be penny wise, pound foolish, well, I was one day washing the pipette tips because I was trained here in UM, and during my honors year project, I was told to wash the pipette tips because they're expensive. You have to recycle, and my supervisor told me, oh, you can actually, I've been keeping some of the pipettes for 7 years. Don't waste any of them, don't throw them. So in Singapore, these are brand new, so I kept washing them, and he told me, why are you washing the pipette tips? I said, they're new. And he told me, well, in Singapore, water is more expensive than the pipette tips. Please throw them, don't wash, don't waste water. So, and as for the third part, don't waste taxpayers' money. I was always frugal because UM trained me to be frugal. And it's usually the guy who got this statement, because he always forgot to feed his animals, because he was doing bioavailability studies. He always forgot to feed the animals and they died. So it was a waste. He had to start all over again with the experiment. So that was that. And of course, another 3 main questions. He will walk around and ask, is your work novel? Because he always gave us the freedom to design our experiments. So next question is it publishable? So when you confirm that your work is novel and it is publishable, then next he'll ask us, when can you give me the draft, meaning the manuscript draft? So what I'm trying to tell you here is, in Singapore, more than 20 years ago, the publication culture and the push for publication was already in place. And with that, of course, with the push, I managed to get about 6 papers out of my PhD. And this paper particularly, in fact, you'll be surprised, my first paper was actually submitted, or first manuscript was submitted to Nature. And of course, since those days, no emails and so on, so I didn't get a 24-hour rejection, but 2 weeks later, I got a very polite rejection. But anyway, I did get some decent papers. They were all like tier 1 publications. And I'm very happy and proud of these papers. Okay, after my convocation in 1994 and 96, I joined UM, and in the interim period, I actually had a stint at working in Inti College, as well as IMC, which is now currently known as IMU. Now, after coming back to UM, of course, because well, people have asked, why did you leave IMU? Or rather, in those days, it was known as IMC. It was a higher paid job for a lower paid job. So I said, I want to do research. But after coming here, and at this point I must also mention that my supervisor passed away 2 weeks after my convocation. So I was left with no mentor. When I came back to Malaysia, I realized that hardly anyone was working in this field. So, or at least even if they were, there was no reflection in terms of the publications. So I had to think and finally decided to work on diabetes, and looked at both aspects, the oxidant aspect and as well as the antioxidant aspect of it. So why diabetes? Because in 1996, the prevalence, we considered the prevalence as very high. It was 8.3. But now we know the prevalence is 17.5, which is not just high, but it's alarming. So most of the diabetic patients in Malaysia are of the type 2 variety. About 95% of them are with type 2 diabetes. And type 2 diabetes is basically related to insulin resistance. In other words, there's no efficient uptake of glucose into the cell because of the failure of appropriate insulin transduction, signal transduction. And so I asked these questions: could high glucose level affect oxidant-antioxidant system? Why or how does high blood glucose level lead to diabetic complications? Are there any ethnic differences in terms of susceptibility to oxidative stress? And do exogenous antioxidants mitigate oxidative stress? If they do, what are the mechanisms? So with these in mind, and with some hypothesis which was already in existence but needed more confirmation to support the hypothesis, we carried on with design more experiments. But meanwhile, let me just explain to you the hypothesis. So when you have hyperglycemia, in other words, high level of glucose in your blood circulation, the mitochondria produces more reactive oxygen and reactive nitrogen species. You know, as I explained earlier, these are a mixture of the oxidants as well as the free radicals, which can collectively cause activation of various biochemical pathways, including protein glycation. So, and that will lead to antioxidants level to be reduced. So here, when I talk about antioxidant, predominantly they are the enzymic antioxidants. Enzymes are proteins, and because of the hyperglycemic state, they could be glycated. Once the enzyme proteins are glycated, they can become inactive. Now, collectively, this will lead to oxidative stress, meaning that the damage, or rather the free radicals or the oxidants that cause damage to macromolecules are much more than the antioxidants. So, of course, with the excessive damage, depending on where, you may have macrovascular or microvascular damages, which includes your heart disease, nephropathy, neuropathy, and also retinopathy, the more common ones. So now coming back to what I did after coming back to UM, well, I had, my first grant was 8,000 ringgit, which was the Board F grant, which I applied within the first week of joining UM. And within the following 1 or 2 months, I got the result. I got the 8,000 ringgit. And with that, I started to set up my antioxidant system, or rather the assay system. And that was the preliminary work. So I used to obtain expired blood. I worked on expired blood, but of course not very old expired blood, because those would be hemolyzed. So this would be like newly expired, just 1 or 2 days old. And I used to go to the blood bank to collect them regularly. And so much so, the person in charge asked, have you turned into a vampire? Every week you're coming over to get blood. But anyway, I needed the blood so that I can test all these assays to see whether they work. And after setting up successfully, and after subsequent ERPA grant, I was able to find clinical collaborators to work on my patient samples. And what I discovered was, yes, there was presence of type 2 DM, or diabetic mellitus related oxidative stress among our Malaysians. And there was certainly age and ethnic variations in the oxidative stress among the Malaysian patients. And there was something interesting here. We found that the Malaysian Indians with diabetes have the highest level of oxidative stress and also have increased incidence of the complications. Now, we were the first to report throughout the world that xanthine oxidase is elevated in that type 2 diabetes. And of course, there were no racial difference, ethnic difference, because the Malay, Chinese, and Indian, they had elevated level, but in comparison to the control, this was very, very significant. Now, this enzyme is actually a purine metabolizing enzyme, but along the process, it actually produces superoxide, which is a radical, and this can cause damage. So we confirmed that this could actually contribute to the damage that happens in hyperglycemia. And of course, in diabetic nephropathy, we found that the contribution of components in the pathology of renal failure towards oxidative stress exceeds that of hyperglycemia. And we showed that diabetes mellitus exacerbates or enhances the advanced glycation end product accumulation in the veins of end-stage renal failure patients, in short ESRD patients. And also there was an association between plasma soluble Raj, or rather receptor for advanced glycated end products, and renal function, which is unaffected by medication usage, and also the enzymatic antioxidants, meaning the superoxide dismutase, catalase, and GPX, or glutathione peroxidase, in chronic kidney disease with type 2 diabetes. So for the diabetic retinopathy, we confirm that the certain polymorphism, okay, of the receptor for the advanced glycated end products gene is associated with diabetic retinopathy in the Malaysian population. We also showed that there was attenuated levels of pro-inflammatory markers in diabetic retinopathy patients undergoing treatment with anti-hyperglycemic and anti-hypertensive drugs. So these drugs, to a certain extent, help to reduce the pro-inflammation. And of course, we also showed that soluble receptor for advanced glycation end product is similar to the receptor that I mentioned earlier, but this one is in the soluble form, is found in the blood circulation. And versus the pentosidine. Pentosidine is actually a marker for advanced glycation end product. So the ratio can serve as a potential risk factor determinant for type 2 diabetic retinopathy. So now, it will not be fair if I don't mention all the other diseases that I ventured into or tested for the oxidative stress and some of the theories and hypotheses there. So we actually tested some lifestyle diseases, made comparisons with some lifestyle diseases, hereditary disease, particularly the beta thalassemia major, and also infection. Basically, we looked at the blastocystis hominis infection. So we actually published quite a lot of papers based on these diseases, but I'll just give you a highlights or something interesting that I noted with these publications. So like in terms of lifestyle diseases, we found that oxidative damage to protein is significantly higher in cancer than hypertension and diabetes. And we found that in thalassemia patients have to be on regular blood transfusion. So when you're on regular blood transfusion, you have a tendency to accumulate iron. Okay, and when iron, like I mentioned earlier on, they can participate in Fenton-Lebowitz reaction to enhance or amplify the radical formation, free radical formation. So therefore, they can actually cause extensive damage, cellular damage. So by having a chelation therapy, in other words, this drug, desperioxamine, actually help to chelate the iron, and therefore your oxidative damage can be attenuated, and we found that the total antioxidant level were also elevated. And as for the blastocystis, it's actually a kind of parasite. Oxidative stress exacerbated parasitic infection, and vice versa. Parasitic infection can also cause oxidative stress in this case. We tested using both animal model as well as cellular model, and apart from that, it caused increase in inflammation, and also we found that it predisposes to colorectal cancer. So these were some of the interesting highlights. And collectively, there was one common factor which we helped to unravel in terms of the Enigma of oxidant-antioxidant balance. We found that oxidative stress has got close relationship with the inflammation. Okay, inflammation can also lead to oxidative stress and vice versa. And now the next part of my journey is the unveiling the mechanism of action of compounds from natural sources with antioxidant activities. My focus is still on diabetes and related complications, as well as to look into the mitigation of oxidative stress. So I use both cellular model and animal model, as well as many of the in vitro, a battery of in vitro assays. So I focused on both plants and mushrooms. Thanks to Prof. Vicky, so I had quite a lot of mushrooms to work on. And apart from that, by then with more grants, I was able to purchase the expensive flavonoids as well. And I did try my hand at some of the trace elements because these were important prosthetic groups or cofactors for the antioxidant enzymes. So it would be interesting. So I had actually worked on all these compounds and extracts. But however, I won't have time to discuss all that I've done. So I'll just take one example, which is the mushroom. So what we did in the in vitro and in vivo pertaining to the diabetes and obesity. So this mushroom is Pleurotus species, or Pleurotus sajukaju, and this is also, common name is known as grey oyster mushroom. If you go to the supermarket, you can get this. It's very common and very delicious. And extracted the beta-glucan-rich polysaccharides from this, and I'll abbreviate now on, I'll refer to it as this, PSCG. And we carried out in vitro assessment and in vivo assessment. Our in vitro assessment was based on the 3T3L1 cells, which are the adipocyte cells, pre-adipocytes, and also the C57BL6J mice, these are genetically modified mice. And we carried out a battery of assays for both studies. And I won't bore you with all the graphs and figures, I'll just give you the highlight. So the extract showed insulin-like activity, which was very exciting. And exciting why? Because it showed, just like insulin, it showed increase in differentiation of the pre-adipocytes, and it caused lipogenesis. In other words, it drove the lipid accumulation, just like what insulin does. And it also stimulated the glucose uptake, and it was via the GLUT4 transporter, okay, just like what insulin does. And also it increased the expression of adiponectin and leptin. Mind you, these are all based on the cellular assays or cell-based assays. And this is also similar to what insulin does. However, in fully differentiated adipocytes, in other words, the lipids are already full, it's accumulated, means you have already primed it to adipocytes, fully mature adipocytes. So when you incubate the extract with these mature adipocytes, what happens is it doesn't behave like insulin. Instead, it helps to mobilize the accumulated fat. So this behavior, and it is through the AMPK pathway, which is similar to a beta-adrenergic receptor mediated action. Furthermore, it attenuated oxidative damage. How we determined this was by measuring the lipid hydroperoxide as well as the protein carbonyl content. These are actually the measurements, the product that we can measure to represent the damage to fat as well as damage to the protein. Now, for the in vivo aspect. So, we actually designed our experiments so that we divided the animals to several groups. We have the normal diet, which comprises about 5% fat, and also high fat diet, which has got about 45% or 60%. So the first 3 weeks we fed 45% high fat diet, and subsequent weeks we gave about 60% fat. So a total of 15 weeks. And we administered, of course, some groups had, or rather we administered the extract, some groups we did not. So we only administered 3 times a week, not every day. And the positive control, we use metformin. Metformin is a common anti-diabetic drug. So what we found was the extract prevented excess weight gain in high-fat diet fat animals. And it caused, high-fat diet caused hyperlipidemia, and the groups which were treated with this extract remained normal. So when you say hyperlipidemia, it means total cholesterol was elevated, triglyceride was elevated, LDL cholesterol was elevated, but HDL was reduced. And we also found that high-fat diet caused insulin resistance, and actually the animals developed diabetes. So we could classify these as type 2 diabetes because it came together with the insulin resistance. And our extract-treated animals remained normal. And also high-fat diet caused increased oxidative damage, and our extract attenuated the damage. So we managed to obtain 2 patents of this work, from this work. More than the patents, I was excited about the fact that our in vitro assessment and the in vivo assessment concurred. Why? Because the in vitro assessment system seemed to serve as a good screening tool, because there are so many natural products that you want to screen, and this will be a quick, more humane way of screening, so that you don't sacrifice too many animals. Okay, I had enough of working on animals during my PhD days. I used to cry after administering venom to the animals, and so I was like always trying to find ways to minimize the usage of animals. Anyway, when times are bad, when you don't have enough funding, which has happened, it's a cycle of like every 5 years or 10 years you'll come across that situation. I decided to do some cooking. Okay, I cooked vegetables. So to look at the antioxidant, because then you can relate to the general public. So what we found was boiling or microwaving improved the antioxidant activity in selected vegetables. While pressure cooking, because mind you, when you say pressure cook, it's like you will find that the color changes and the color of the vegetable will change, and there won't be, you'll think that all the nutritional content is gone, there's nothing there left. But actually the antioxidant level did not really reduce that much. Okay, so there was no significant decline in the antioxidant property. And very interestingly, when you cook for a very short period, especially boiling or microwaving for just below 5 minutes, for example, your vitamin C level can actually be higher than the raw. You always think that when you heat treat something, the vegetables, the vitamin C will be destroyed. But what we found was the vitamin C is elevated. However, if you prolong the cooking period, then the vitamin C is destroyed. So we tried to find an explanation. So one of the explanations for the total antioxidant elevation is probably because of the matrix softening of the material, the plant. Therefore it allows the phytochemicals to be accessible for the assays. Now meanwhile, for vitamin C, we thought probably the short duration, the heat treatment, destroys the enzyme that is involved in inactivating the vitamin C. So by doing that, your vitamin C remains active. So probably that's the reason. So, however, I was not happy with just this explanation. So I tried to venture into 2D gel electrophoresis, and we used the bitter gourd. So among the plants that I mentioned earlier, the vegetables that I mentioned, I forgot to state earlier, it's bitter gourd, kangkong, long beans, and broccoli. And here we just focused on bitter gourd as an example. So what we did was the heat-treated vegetables, the extracts, were subjected to 2D gel electrophoresis. Now the student who was asked to do this assay thought I was crazy, because he said, why are you have already heat-treated the extract, the proteins will be destroyed with heat. And why would you want to do a 2D gel electrophoresis? You'll only see streaks of imager that is all streaking proteins and there's nothing there. I said, what about heat shock? I've heard of heat shock proteins. There may be heat shock proteins. So never mind, you go ahead and do it. And he did it. So a very good, diligent student. And we found that there were certain stress response proteins which was expressed. And of course, we didn't use the pressure cooked sample because they were definitely, all the proteins were destroyed. For the 5 minutes boiling and microwaving, yes, there were very distinct proteins which were expressed. And among these were the superoxide dismutase and few other heat shock proteins. So we speculated that these could actually help to protect the phytochemicals. So the antioxidant properties of these phytochemicals probably were retained that way. So now the next question, does prevention or mitigation of oxidative stress depend on strong exogenous antioxidants? Well, you know that some extracts, based on our experience, we found that some extracts are compounds with weak antioxidant activity, especially as measured in vitro, seem to have high potency to mitigate oxidative stress in vivo. I mentioned to you that I've worked on both plants and mushrooms. So generally the mushrooms have got lower antioxidant property compared to the plants. The plants were very much higher compared to the mushrooms. And yet, in the in vivo system, we found that many of these mushrooms were very, very potent and very good at mitigating the oxidative stress. So this shows that it's not just the exogenous antioxidants, the phytochemicals, the vitamin C and A and so on, which is contributing to the antioxidant property in terms of mitigation of the oxidative stress, but probably the endogenous antioxidants, which are the enzymes. And these enzymes are proteins. All right, so we don't really need that high potent or high potency antioxidants. That's what my opinion is. And we also carried out some experiments to show whether there's any association between the antioxidant level and the damage to DNA in mononuclear cells. And we found, in this study, we actually screened through about 20 different edible plants and herbs, and we found that there was a strong association between the level of antioxidant and as well as the genotoxic property. So this is actually a single cell gel electrophoresis image. This is the intact DNA and this is damage. So particularly among these plants, there were two. Quercus infectoria, I think in Malay it's known as manjakani, and also Vitex pinnata. I forgot the Malay name for this. But these two had very high antioxidant potential or property based on the assays that we carried out. And when we tested on the DNA, we ran a single gel electrophoresis after exposing to the mononuclear cells, and we found that there was very significant damage. Okay. So now, coming back to the common question that most people ask, antioxidant supplements, do they really work? Because now, there are so many people working on antioxidant, especially in Malaysia and Southeast Asia particularly, in Indonesia, in Thailand. And after doing some in vitro work, they are very quick to market it as antioxidant supplements. So do they really work? Do we really need them? Or are they a recipe for disaster? So before we determine that, we have to consider that there's a possibility that some of these supplements can cause endocrine disruption. So when I say the endocrine disruption, usually hormone binds to the receptor on the cell membrane, and after that you have your signal transduction for the relative, or rather the appropriate biological response. But these supplements, some of these molecules may actually bind to the receptor instead, and they may actually cause a disruption of the normal hormone function. So to give an example, the compound called genistein, which is a kind of flavonoid from soya bean, at one point of time it was like marketed quite widely, and now it's banned. Because just to get 1 milligram of that genistein, probably you have to extract from a few kilos of soybean. So now, soybean is good. If you eat it, it's a good source of protein and other phytochemicals, but you are not going to eat like 1 or 2 kilos of soya in a day. Whereas these supplements come in a few milligrams. All right, so when you have taken it, so the phytochemical that goes into your body is really mega dose. So naturally you'll have the consequence, all right? So, and as for genotoxicity, just like what I've shown earlier, there's a potential that it can actually, high antioxidant level can cause damage to the DNA. And of course, next question, the bioavailability. We don't know whether the supplements that you consume, whether they actually are able to pass through the gut into the bloodstream. Okay, that's one question. Next, there's a possibility that they may actually interfere with micronutrient absorption, means other micronutrients which are essential, including your other vitamins and also some minerals. So they could actually impact. So not much is known. A lot of studies need to be done. We know about vitamins and some of the minerals because we have known since many, many decades. But all these phytochemicals, we are out in the market and people are actually consuming quite in large quantities. And of course, next is the influence on gut microbiota. So more recent studies have shown that the composition of your bacteria in the gut actually have an influence on how your body accumulates fat, and in fact it's also linked to many kinds of diseases and your health status. So if you're going to disturb this balance within the gut, perhaps some of these supplements might wipe off the good bacteria, or alternatively it might wipe off the bad bacteria and may retain the good bacteria. We don't know. There's a lot to be looked into. All right, so that might actually cause an influence on how your body functions or your normal homeostasis. So right now, we're still back to consuming antioxidant should still be along with a balanced diet for optimal benefit. That will be the safest bet, because I can't say, standing here, I can't say that the high antioxidant supplement is going to be useful. Okay, and in summary, oxidative stress is associated with disease state, and this may be mitigated by achieving a balance between oxidants and antioxidants. Oxidative stress and inflammation are interrelated, and it is futile and may be detrimental to consume high-dose antioxidant supplements, so be careful. And it is not possible to achieve oxidative balance through diet alone. Now, why I've chosen these colors is just to show that among these fruits and vegetables which are supposed to be rich in antioxidants, you'll find that most of them are related to these colors. So don't eat just one type of color. So don't just take greens. Make sure you have purple, red, orange, and a mixture of all this. There may be different shades, maybe a darker shade or a lighter shade and so on. Anyway, bear in mind, diet is only one part of the whole scenario. For sound health, and in order to achieve your oxidative balance, you need to drink plenty of water, sleep well, avoid or manage stress, be calm. You can do yoga or Tai Chi and so on, or exercise, eat a balanced diet, try to maintain an ideal BMI, and exercise. So science never solves a problem without creating 10 more. Therefore, my journey continues. Okay, so now is the acknowledgement part. So I have to thank all the grant awarding bodies, Universiti Malaya, Ministry of Higher Education, Ministry of Science and Technology, and all my collaborators, particularly Prof. Vicky, Prof. Chua, Prof. Suresh, with whom I've had extensive collaboration, and I've got quite a large number of papers published with them. And of course, among my clinical collaborators, I have to acknowledge Prof. Dr. Rokia Pende, who passed away a few years ago. She was my first clinical collaborator and a long-standing one. Even after a few months after she passed away, we had a paper together, published. And of course, all my external collaborators. And all my students, thank you. And this is Dr. Gauri. In fact, our patented products were actually her work. And she was my PhD student, and for a short period of time, she was also my postdoctoral fellow. Unfortunately, she passed away 2 years ago. And she was a big loss. And here, this is Chai. He was my first PhD student. And he's here right now. And these are all my students. And I would like to say thank you to all of them. And also, I'd like to mention here that I also learned a lot from my students. And this is my department. We are more like my family. I'd like to thank the Dean and Faculty of Medicine in general, all the members of the staff, for the support rendered to the department as well as our biomedical science program. This is my family. This is my husband. He's seated over here. Thank you for your love and support. He has always been a strong pillar of strength throughout my career. My parents, my brother and his wife, they have always been there for me. Particularly my brother, he had never said no. He actually, for the booklet, the cover was designed by him. And during my PhD days, I bullied him into typing all my manuscripts because we didn't have Microsoft Word. It was WordPerfect, and I hated using that. I didn't know how to use it. And I used to come back home over the weekends, and he was in school then. He was just a schoolboy, secondary school boy, and yet he actually, for the sister's sake, he actually typed all my manuscripts. And even when he was doing his STPM, he helped me to type my thesis. So he saw me typing with one finger and he said, "Come, let me do it for you for a while." So, and my children, my niece and nephew, they have always kept me on my toes and ensured that my life was exciting and lively. And just some from my album, some photos from my album. Okay, this is the networking. And the future direction for biomedical science, and proud moments with our biomedical science undergraduates. So this particular symposium was very memorable because of all the categories, oral presentation by our students. They won all the categories. So it was a very proud moment for me. And I'm not a great inventor because I've always been passionate about fundamental research, but this is my own small way of contributing to innovation. And of course, this is some of the activities with the Women's Federation for World Peace. Thank you for your attention.
Thank you, Professor Umah, for sharing your journey with us and for sharing your knowledge with us. We now invite Professor Adiba to conclude the lecture, which has just been delivered by Professor Umah.
Thank you, Professor Umah, for a wonderful lecture that took me back to my early days in biochemistry. And it takes a great teacher to make biochemistry come alive and interesting as you did today. And I think for the non-scientists in the audience, relatively easy to follow and understand, I'm sure. And it takes a great teacher to inspire so many masters and PhD candidates. And I think you've also probably helped many of us to save a lot of money from buying all those supplements. I don't know how many of you actually buy supplements. I don't, but I guess you've kind of reminded us again to live a healthy, long and healthy life. Unfortunately, it still takes a lot of hard work of exercising, eating the right things, and not just a matter of popping pills. But the real message that came through to me from your lecture today, apart from all that I've just said, is also your passion, as you said, for the fundamental science that helps to answer many of these questions that many people just assume. For instance, in terms of the antioxidants and how you could possibly solve so many problems just from taking multiple colored pills, and there are enough people out there willing to take advantage of our vulnerability and our wish to remain young forever. So I think it's wonderful that you've persevered to kind of debunk some of these myths. But it also takes a brave soul to come out to debunk this, because I'm sure it's a multi-billion-dollar industry that will not appreciate some of the science that you have tried to disprove. And finally, I think it also takes a great scientist to be creative in lean times, to even think about microwaves and boiling pots and its effects on food, and still be able to publish in very good journals. So there you go. So money is not always the answer, but it forces you to become very creative, and I was very impressed with how you undertook that in lean times. So there will be more papers, I'm sure, in the next few years from microwaves of Professor Umah Rani in the Department of Biochemistry. So with that, ladies and gentlemen, thank you very much, and once again, congratulations.
Thank you, Professor Adiba, for chairing this session. And right now, I'd just like to take a quick moment for Professor Umah's students to show their appreciation. We have Dr. Bhavani and Mr. Wong Fu Nien from Professor Umah's Biochemistry Lab. And we have Professor Marianne Tan, very good friend of Professor Umah, also presenting her with a gift. Thank you again to all. Thank you, Professor Umah. Thank you, Professor Adiba and management of the Faculty of Medicine. Thank you to UM staff and students who have attended. And thank you to all guests here, including Professor Umah's family. We have reached the end of our session. And on behalf of the University of Malaya, I would like to apologize if there's been any weaknesses or shortcomings in the organization of this lecture. But now we have food, so please do join us at Synapse upstairs, and there's food as well outside DK1. So thank you again, everybody.